We know that to pass the hereditary information to the progeny, any given cell needs to replicate its genome. The DNA replication is therefore one of the most fundamental processes in biology. Today, we are going to look at this mechanism in cells. And we already encountered the first central enzyme, the DNA polymerase III, that does the DNA replication in E. coli. We also looked at the chemistry of the DNA synthesis, and we noted that the chain elongation occurs unidirectionally from the 5' to 3' end. So let's go back to our simple building blocks and look at this one more time. So I need a mother template that I want to replicate, right? And in order to get this done, I need the DNA polymerase. But you also see that I have a short stretch of what it's called the primer. Indeed, there are universal facts about DNA polymerases, and the direction of synthesis we already looked at, 5' to 3', and DNA polymerase always needs a primer to start the DNA synthesis. You may have already noticed that we keep looking at this, let's say, the convenient strand of the parental double helix, the S' strand, where we can sit on top and do the straightforward synthesis from 5' to 3'. Well, if the replication machinery, if the polymerase were to go unidirectionally to synthesize the DNA, then we have a problem because the complementary strand, the S strand, has exactly the wrong direction. How does nature deal with that? Well, it turns out, as always, it has an ingenious solution. What it does, it synthesizes this so-called leading strand in a straightforward fashion and deals with the wrong directionality in a way that it still synthesizes, of course, in the right direction, but instead of going through, of course, it makes small so-called Okazaki fragments and stitches them together at the very end. So, as you see here, the DNA polymerase III is more or less symmetric. So, one half works on the leading strand, the other half works on the lagging strand. Before we deal with the intricacies of this DNA synthesis, let's take a look at the DNA itself. There are indeed big differences between eukaryotes and prokaryotes. One big difference we already know, the eukaryotic DNA is stuffed into the nucleus, prokaryotic DNA is exposed to the cytoplasm. Another big difference is that prokaryotes have circular DNA. E. coli, for instance, has a circular DNA with a very specific replication origin that signals to the replication machinery to land there and to start the synthesis. It looks quite different in our cells. So first, we have linear DNA, and second, we have many origin of replication, and they are really not easy to spot. Remember, E. coli has about 4.5 million base pairs, and using these 4 letters, it has created a word, if you will, that is recognized by this replication machinery among all these 4.5 million letters, which is quite impressive right there. And it turns out to be a word with about 250 base pairs, and people have already decoded this sequence. It turns out to be GATC, but this alone wouldn't work because we would encounter this particular sequence, statistically speaking, often in the genome. So what it does, it just makes a tandem array of this consensus sequence, as illustrated here with the green boxes. And the consensus sequence has a lot of A's and T's, and this is because at this position, the double-stranded DNA needs to open up in order to start the synthesis. So having 2 hydrogen bonds is more advantageous than having C and G and 3 hydrogen bonds. And lastly, we see the binding sites for the DNA A protein. So this protein now recruits our first machine, the helicase. And here you see the remarkable structure of this hexameric helicase. What this does is it uses ATP, the real ATP, as a fuel in the cell, and not the DATP, the oxy, that is now being incorporated into this polymer. So basically what it does is it just flows through the double helix, makes single-stranded DNA, but that's a problem because single-stranded DNA is unstable, can get attacked, plus it could also form secondary structures depending on the base pairs. So, solution, nature has then thought about single strand binding proteins. So those are the ones that recognize specifically the single strand and start lining up, protecting the whole thing and making the life of the polymerase later a lot easier. Now we opened up the double-stranded DNA and can start the synthesis. So the so-called leading strand will be straightforward. It will be synthesized from 5 to 3 prime, but the complementary strand will be more difficult if the machinery wants to go unidirectionally. So instead of continuously doing the synthesis, it does a bit of a synthesis in the right direction, the so-called Okazaki fragments. The problem is, of course, if you do not do the continuous synthesis and have these little pieces, A, you need to stitch them together, and B, each time you start one of these Okazaki fragments, you are going to need a primer. So this is where the DNA primase comes in. It is called DNA primase, but it makes RNA primer. And this is the one that continuously supplies the primer for the Okazaki fragment synthesis. The second problem, we now synthesize these Okazaki fragments. We need to stitch them together. And our next machine, the so-called DNA ligase, that basically at the end of the day seals these phosphodiester bonds between these little Okazaki fragments. So to summarize these steps, what happens is that the DNA primase synthesizes the primer, the RNA primer, so that the DNA polymerase now can come in, do the synthesis, and this goes until it reaches the Okazaki fragment that has been synthesized before. And then this primer needs to be removed and replaced by DNA, and this is being done by DNA polymerase 1, the first polymerase that has been purified by Arthur Kornberg. And the only thing remaining here is the ligation of these pieces, and that's basically the way how nature deals with this lagging strand synthesis. When we put all these things together, we have the complete DNA synthesis machinery, and complete is really a long stretch because I think these days people think that there are over 20 different factors that are incorporated into this replication machinery. When we look at the simplified schematics, then we spot nature's trick in order to get the synthesis direction right for both strands, the leading and the lagging strand. In the next slide, I'm going to show you the machinery in action. So pay close attention how the machinery deals with this lagging strand.
In a replication fork, two DNA polymerases collaborate to copy the leading strand template and the lagging strand template DNA. In this picture, the DNA polymerase that produces the lagging strand has just finished an Okazaki fragment. The clamp that keeps the lower DNA polymerase attached to the lagging strand dissociates, and the DNA polymerase temporarily releases the lagging strand template DNA. As the DNA helicase continues to unwind the parental DNA, the primase becomes activated and synthesizes a short RNA primer on the growing lagging strand. The DNA polymerase binds to the DNA again and becomes locked in by the clamp. The polymerase uses the RNA primer to begin a short copy of the lagging strand template DNA. The polymerase stalls when it reaches the RNA primer of the preceding Okazaki fragment, and the entire cycle repeats.
The movie suggests that the machinery plows through to replicate the DNA. Well, will it be that straightforward? Of course not. Will the polymerase 3 always incorporate the right nucleotide? Of course not. Well, it turns out the machinery can also play Pac-Man. We zoom back into the replication machinery and take a closer look at the polymerase 3. So we know, of course, it has a polymerase activity 5 to 3 prime, but it also has a 3 to 5 prime exonuclease activity, meaning it can reverse the step of the polymerase. Remember, when we looked at the different polymerases in E. coli, we encountered these cryptic sounding descriptions. 5 to 3 prime polymerase activity that we of course know, but we also saw that practically all of those have also the 3 prime 5 prime exonuclease activity. So let's go step by step to understand what this exonuclease activity is. On the left, we see the schematics of the 3 to 5 prime exonuclease activity. So here, the correct base pairing is between the colors blue and yellow. And if the polymerase tries to squeeze in the wrong nucleotide, the 3 to 5 prime exonuclease activity will kick in, meaning it will cleave the phosphodiester bond that has been formed by the polymerase. And when the correct nucleotide comes in, the machinery continues. But this is not happening just one nucleotide by one nucleotide. If something like this happens, the machinery really goes back a couple of steps and continues the synthesis. Here we see the structure of the DNA with its exonuclease domain at the bottom. And when we look at the simplified schematics, this really looks like a hand. And we see that this enzyme combines different catalytic centers. And in one center, it does the synthesis. If something goes wrong, the exonuclease activity takes over, corrects the mistake, and the polymerase can continue. Let's go back to our overview of the replication machinery. We now know that we have a polymerase there that can do the synthesis. It can also proofread. We have the DNA helicase that can flow through the double-stranded DNA to open it up and allow the synthesis. One more machine actually is still missing here. So when the DNA helicase flows through the double-stranded DNA, it will eventually create supercoils, and worst case scenario, this will then stall the whole machinery. The question is now, how does nature deal with this problem? It doesn't matter in what type of cell you are. You can have a prokaryotic cell, you can have a eukaryotic cell. The DNA will not freely float around in the room. So it will be anchored somewhere, somehow. So when the replication machinery pulls forward, what it will do is it will create supercoils in front of it. How can you get rid of them? Because worst case scenario, the whole machinery will stall. You could think, okay, the replication machinery itself could rotate, but this is going to give you a different problem. So you will catenate the DNAs. And the problem here is, if you want to separate during the cell division, well, this is also going to be a huge problem. To solve this particular problem, nature has invented another fascinating machine called topoisomerase. This particular machine can introduce a double-strand cleavage and pass through the second double strand, religate the cleavage, and at the end, we have decatenated the two DNA double strands. On this slide, you see the beautiful structure of the topoisomerase. So it is a homodimer, and as you can imagine, this enzyme is also subject to intense research, and the proposed mechanism that we have today is that it grabs the double-stranded DNA, the first one, the gDNA, then it grabs the tDNA, the transported DNA. The name already suggests that's the one that's going to be transported all the way through the enzyme. And of course, it needs energy, so it hydrolyzes two ATPs, introduces the cleavage in the gDNA, passes through the tDNA, and reseals and releases. If something goes wrong with this enzyme, it really doesn't matter if you are an E. coli cell or any type of human cell. This is going to stall everything and kill the cell. Now we have a list of machines that the cell needs in order to replicate the DNA. So one thing is knowing the mechanisms, etc., etc. The other question is whether or not we can actually benefit from this knowledge. We know that E. coli is everywhere, including our body. And let's say the bad type of E. coli, if it takes over in the intestine and the body cannot fight it successfully, this is going to have some unpleasant consequences. How does our intestine look like? So thank goodness we have electron microscopy. Here we see these cells in the intestine. So very conspicuous are these finger-like protrusions sticking out of the cell surface, and these are filled with the so-called actin filaments. So these are different kind of ropes we can find in our body. And in the next slide, I'm going to show you a movie how an E. coli cell actually infects one of our intestine cells and actually hijacks the actin for its own purpose.
So here's our bacterium here. And behind it will be these long strands that are the bundle-forming pilus. They're going to adhere to the microvilli, these wiener-like structures. This is what your normal intestinal cell surface comes along here. Bacterium then causes these things to disappear. It then starts to nestle down on the surface. It's locked in. And then it's going to come down, and it's going to set up camp right there. We know that happens because we can see this in electron micrographs, and the microvilli all disappear, and boom. Okay, so we're now stuck in the intestine. Bacteria are here. We're going to zoom in. Here's our type 3 secretion system, right? This thing will poke out. We know protein comes out. We know this protein. And then it inserts two more proteins that come out and down this tube, and they go in to insert into the host membrane. So these green proteins will sort of form like a donut pore, and this is kind of the tip of the syringe. Then we have to fire in the receptor, the TIR molecule, and you'll see it as this red molecule coming down the chutes. And it gets then fired into the host cell. And then it goes into the host cell. We don't know if it goes through the cytoplasm, goes straight in the membrane. We haven't figured this out. You'll see a little blue dot. That's a tyrosine phosphorylation event. Bacteria gets modified. Then it ends up in the host membrane. So here we're piling the receptor into the host cell. And then we then have to dock tier with intimate. So we think this tube-like protein retracts. And what will that then do is bring the bacteria down into contact. Here's intimate up here, blue tier here. Houston, we have contact. We have now landed. Bacteria is stuck on this cell. And now we're going to go inside the cell, and we're going to start to see the face of tier that's facing. It recruits cytoskeletal proteins and these little donut-shaped things we are starting to understand more. Then you're going to see little yellow beads come flying in. These are actin monomers. They then get together, and it's like a string of pearls. They polymerize. See these things flying in. They then start to get longer and longer. And actin does this for a living. It forms these long projections, strings of beads. And what that will start to do is start to put a force on this thing, and you will then start to see the bacteria actually raise up on the cell. And it's because there's actin pushing underneath it that it actually pushes it up into the cell. There it goes. And then it starts to push up. Bacteria starts to raise up. It's now building its throne. Then I'm going to sit up on top of this thing and rule out over it. And if this doesn't want to make you be a microbiologist, I don't know what will. Isn't this a thing of beauty? And then it just continues to rise and rise and rise. And the end result is we then end up with the bacteria sitting up on this throne, shall we say, to rule out over the mammalian cell. And that's what we see in disease.
We just watched an E. coli cell invading our intestine. And we also saw that we really do not understand every single step of this infection. But it turns out if you have some decent idea about the machinery that keeps these E. coli cells alive, you can already get quite far. Two of those we already encountered. The first one is the topoisomerase. We can indeed develop substances to specifically inhibit their machinery and not ours. Or we can also go against the DNA polymerase itself, etc. And this is being really done against cancerous cells. And at these examples, you can see how important it is to really A, discover these machines, and B, understand how they work. Now we have learned quite a bit from cells how they replicate their genome. So what we can do now is we can learn from the cell and do this job in the test tube. To understand what this means, let's meet in Yellowstone for the next lecture.